Hello friends, continuation of seed technology basic notes part 3. First seed testing station or lab was established in Germany. That was by Nobbe, that is Frederick Nobbe, father of seed testing is also his another name. In India, the testing station was established in 1961. ISTA was established during 1924 in Zurich, Switzerland. Rules of seed testing by AOSA, that is Association of Official Seed Analysts, is given. CSTL is Central Seed Testing Laboratory, which is located at New Delhi, established during 1960. All seed testing laboratories are established under Seed Act 1966. 140 seed testing labs or authorities are under ISTA. Seed technology, the book or the standard guidelines are given by AOSA, that is Association of Official Seed Analysts. Seed Science and Technology, the magazine is published by ISTA. First seed testing lab was established at Copenhagen, Denmark. Here, for your convenience, I will repeat it once again. First seed testing station was established in Germany during 1869 by Frederick Nobbe, but first seed testing lab was in Copenhagen, Denmark in 1971. Seed certification agency, these will be changing regularly. Coming to National Seed Program during 1975 to 76, working sample for moisture estimation is 4 to 5 gram. In Leguminosae, Cruciferaceae, Brassicaceae, seed without seed coat is inert matter. Cotyledons of peanut, cowpea and soybean are inert matters. Genetic purity test. In different crops, in wheat, NaOH and phenol tests are used to test the purity. KOH bleach test is used in sorghum. Peroxidase test in soybean. Potassium dichromate test in garden pea and fodder pea. HCl test in cucumber and melon. Electrophoresis in many crops, almost all the crops nowadays it is used. Then examination of seedlings. Grow out test for minimum number of plants in test field is 100 and replication minimum 400 plants are taken. Iodine solution is used to separate millet grains from weedy forms. Coming to sample given for grow out test, 1 kg or 1000 gram is given for cotton, groundnut, maize, soybean. Remember, if seed size is big, the uh, number of grains or the weight of seed will also be more. 500 grams is given in case of sorghum, wheat and rice. 100 grams in case of bajra and jute. 250 tubes or roots or corms given in case of potato, sweet potato, etc. Coming to seed viability test. Seed is the living entity in that embryo, embryo and aileron layer are stained. In case of tetrazoleum test, as I already told, the process was given by Lacon. Here, red compound produced is formazole, which will give the viability index. Working sample taken here is 100 to 200 gram in 4 replications. Embryo should be exposed during testing and therefore remove the seed coat. TZ test can be used both for viability and vigor test, that is intensity of red color is directly proportional to vigor. Example, in case of cereals, Aluron TZ test. Lactophenol solution is used to remove the seed coat, that is used prior to the tetrazoleum test. Indigo carmine test is to check the viability of trees and shrub seeds. Radiography is used to detect damage, empty, polyembryonic seeds. Embryo excision test. Then GADA test is for glutamic acid dicarboxylase activity test. This is how it is made means, here glutamic acid decarboxylase plus viable seed if we'll take, then carbon dioxide concentration will be increased. Carbon dioxide percent is directly proportional to viability. This is used in monocots like wheat, rice, paddy, cotton, soybean. Ferric chloride test for legumes. This is how it is used, means here injured seeds turn black and normal seeds will be intact and normal. EC test for testing seed lichens by injured seeds. This EC is inversely proportional to viability. Seed crushing test is used in jute. Accelerated aging test in soybean. Remember all these tests because in one or the other way they'll ask these questions. Coming to germination test, for most of the crops between paper test is used. For tobacco, cabbage, cauliflower and sesamum, top of paper test. Seed index is weight of 100 seeds and test weight is 1000 seeds. Generally seed index is for bold seeds and test weight is taken for small seeds. Coming to seed vigor test, here physical test is used for size, test weight and soundness. Performance test is for speed of germination and seedling growth rate. Accelerated aging test for allowing to germinate after aging like in soybean. Brick gravel test given by Hiltner in cotton. Paper piercing test by Fritz. Cold test for maize, cotton, soybean. Cool germination test for cotton. Hot water test for maize. Respiratory quotient for rice and maize. Here respiratory quotient is equal to carbon dioxide evolved divided by carbon dioxide consumed, which is directly proportional to vigor. Last is mitochondrial activity test. This is for groundnut. The respiratory quotient is represented or measured by Warburg respirometer. Remember, respiratory quotient is measured with Warburg respirometer. Coming to different classes of seeds and their purity, nucleus seed and breeder seed should be 100% pure, whereas foundation seed should be 99% pure. Here the certification or caretaker of these seeds are also given here. Nucleus and breeder by breeder himself, foundation by NSC, SSC, SAU, certification seed by National Seed Corporation. Here 98% of genetic purity is required for varieties. SSC and companies will also take care of certified seeds. 95% of genetic purity is required in case of hybrids. In cotton, these are all germination tests. Coming to tuber indexing is done in potato to get viral free plants. Coming to seed health tests, there are 4 tests. First one is blotting paper, then agar plating, bioassay, then examination of plant. Mechanical seed divider, here conical divider or Borner divider, that is based on gravity. Riffle seed divider, gametic divider, that is for principle of gametic divider for centrifugation principle it works with. Then in case of here this is all about seed act, control order and all, whichever we have not discussed already. I'll just tell you. The seed control order came in 1983 February 24th. International seed year is 1961. Seed certification is done only to notified varieties. Remember, certification is done only for notified varieties. Seed as essential commodity under Essential Commodity Act 1955, and this is also recommended in Seed Control Order 1983. One state seed certification agency for each state under Section 8. National Seed Corporation established in 1963 March. Seed drying. Seed moisture more than 18% means maximum drying temperature should be 32 degrees Celsius. If less than 18% means 40 degrees Celsius is the maximum seed temperature. In general, drying temperature maximum is 40 degree Celsius. Okay. So somewhere here we lost. Okay. The desiccants used are CaCl2 or calcium chloride, silica gel, phosphorus pentoxide. For seed storage, short term storage is 6 months, seed moisture for drying. Medium term is 18 to 36 months, and long term drying is 5 to 50 years. Seed moisture is less than 14% for starchy seeds and less than 11% for oily seeds. Coming to Harrington's thumb rule, here for every 5 degrees Celsius temperature decrease, shelf life doubles. For every 1% seed moisture decrease, again shelf life doubles. That means between 5 degrees Celsius decrease means the shelf life doubles, and 1% moisture decrease means shelf life doubles. But this rule is applicable only up to a certain level. Coming to good storage, here temperature plus humidity plays very important role. High yielding varietal program started during 65 and 66. Started variety HD 2329 wheat. Seed testing laboratories are established under Section 4 and minimum seed standards are prescribed under Section 6. You should also remember these sections, Section 5, 8, 4, 6. These are very important. New varieties are tested for enders. Lyophilization is freezing in vacuum. Coming to seed structure, endosperm is 3N. Alluron layer is 3N. Seed coat is 2N. That is haploid, diploid, triploid condition. Dead part of seed is endosperm and seed coat. Live part is embryo, as you know. Cotyledon of monocot is scutellum. Secondary roots develop from mesocotyle. Coriander is trimonoecious, where male, female and hermaphrodite flowers are present in single plant. Okay. Pollination between flowers of same plant is geitonogamy. Flowers of different plant is xenogamy. Here see terminologies for pollinators, again it repeated, but I will read because here more number of examples are given. Myrmecophore is the pollination through ants. Zoochory is through animals done in apple and walnut. Anemophily through wind in maize and bajra. Hydrophily through water in rice and pepper. Entomophily through insects. Sunflower and cotton are the examples. Waspophily is through wasps in some tree species. Like pig and all, this is found. Carthrophily through beetles. Melithophily through bees in case of sunflower, safflower and all attractive flowers. Myophily through flies in mango. Psychrophily through butterflies, this is also found in most of the flowering plants. Ornithophily through birds. Chiropterophily through bats. Melassophily through snails and opiophily through snake, that is found in Colocasia. Tripping is the mechanism of honeybee pollination in Lionaceae. Tripping is practiced in Lucerne. Then in case of gymnosperms, double fertilization is absent. Here endosperm is N. Generally in case of angiosperms, endosperm will be in 3N condition, whereas here in gymnosperms it is N. Always remember. Coming to germination, imbibition of water through micropyle is nothing but germination. After seed development and maturation, funicle desiccates, leaving scare on seed coat called hilum. GA3 induces alpha amylase from alluron layer of scutellum. Radical emerges first. Except in waterlogging condition, coleoptile emerges first. Example in case of rice. Okay. Generally, radical emerges first. Quiescent seed means seed which can't germinate even though it is viable. ISST is Indian Society of Seed Technology, established during 1971. Usually working sample is 25 grams and number of seeds is 2500. Diaphanoscope is used for purity analysis in seeds. Nuclear and breeder seed production in wheat is done through mostly ear to row method. For most cereals also this method is used. Tuber indexing in case of potato to test viral disease. To enhance pollination in paddy, rope pulling, flag leaf clipping, GA3 spray. Gametocides used in paddy are Mg1, Mg2 as gibberellic acid source, ethyl, NAA and malic hydrazide. Hybrid seed production in cotton used Doak's method, that is hand emasculation. Coming to synchronization of flowering, which is done through jerking in bajra, staggered planting in maize, N2 application and irradiation in early varieties of sorghum, pearl millet. Irrigation in early variety of sorghum and pearl millet. Only in maize, isolation distance can be altered based on number of border rows and different times of planting, means spatio-temporal isolation is practiced in case of maize. Halm cutting is an important operation in potato, that is cutting off the halm or upper portion of seedlings before harvesting. Okay. Coming to maximum moisture of different crops for storage: rice 13%, cereals 12%, castor 8%, pulses 9% and oilseeds 8 to 9%. Barrier isolation is followed in self-pollinated crops. Initial varietal or hybrid evaluation trials is for 1 year in small areas. Advanced varietal or hybrid evaluation trials for 2 years in multi-locations. Coming to steps of varietal release: evaluation, identification, release and notification. This is a simple step. When I was explaining the varietal release chart or the video which is having elaborated way of varietal release, this simple form is not given, but in majority of the exams they will ask the simplest form only. This you should remember very properly: evaluation, identification, release and notification. This you just fix it in your mind. Notification is done both for variety and crop by Central Seed Committee. Okay. Coming to seed legislation, again I am emphasizing on legislations. These sections are very much important. The first section is section 3, that is for Central Seed Committee or Central Seed Certification Board. Central Seed Certification Board is also comes under section that is CSCB. Section 4 is central and state and central seed testing laboratories. Means these sections talk about the establishment and functioning of particular committees. Okay. Section 5 is for notification of kind or of kind of variety. Section 6 is for labeling compulsory or minimum seed standards. Section 8 is for establishment of seed certification agency. Section 15 is power of seed inspector and section 13 is appointment of seed inspector. Under section 2, jute seed is induced as planting material, that is amended. Coming to xenia and metaxenia concept. Xenia is effect of pollen on seed coat or endosperm. Metaxenia is effect of pollen on seed embryo, example in case of maize. Seed certification first began in Sweden. For storage of foundation seed, 25% humidity and 30% 30 degrees Celsius temperature. For germplasm storage, 5 to 10 degrees degree temperature, 20 to 25% moisture, 4 to 5% 20 to 25% humidity and 4 to 5% moisture is required for storing the germplasm. Coming to different kinds of proteins, in rice, orezine is present, wheat, gluten, oat, globulin, maize, zein. Compact area approach for hybrid seed production: in sunflower and tomato, Ranebennur; cotton, Surat; sorghum, Nandyal, which is in Maharashtra; cauliflower, Solan Valley, which is in Himachal Pradesh. This is also very important. Then off-season nursery area is also very important. Okay. Coming to different fathers. Father of seed testing is Frederick Nobbe. He has written the book Handbook of Seed Testing. Father of seed pathology is Paul Niergaard. Father of Indian seed pathology is Surya Narayanam. Father of Indian seed testing is Amir Sanghi. Father of seed technology Badrinarayan Barwale. Nitrogen positive crops are having the opportunity of nitrogen advances in flowering, like in sorghum and pearl millet. Then phosphorus, that is application of phosphorus advances flowering in case of rice. Maize first hybrid is Ganga 101. Then Ganga 102, Deccan and all those were hybrids came. In PGMS or TGMS, that is for photoperiod or temperature sensitive male sterility, only A and R lines are involved, that is two-line breeding system. In rice, barrier isolation is followed by planting maize, sugarcane, sun hemp. Nitrogen application delays the flowering. That means rice is nitrogen negative. P application or phosphorus application and irrigation advances flowering in case of rice. Red gram ICPH 8. Why only specific points are given here? Because these are frequently asked in different exams. Okay, ICPH 8, the parentage is MS Prabhat DT into ICPL 161. Cotton H4, the parentage is Gujarat 67 into American Nectarless. Ethylene stimulated for ripening, bud dormancy and seed germination. Coming to different objectionable weeds in different crops. In rapeseed and mustard, Argemone mexicana, Berseem, Palak and Shikori, Lucerne, Dodder or Cuscuta. Here see, Berseem is king of fodders and Lucerne is queen of fodders. In Berseem, Shikori and Palak are the major objectionable weeds, whereas in Lucerne, Dodder and Cuscuta are the major objective weeds. Rice, Echinoclova, wheat, Chinopodium. CMS was first found in onion by Jones and Davis. CMS used in paddy, sorghum, sunflower. Unit of seed certification is 25 acres per or 10 hectares. India is not a member of AOSCA. Remember, first seed testing laboratory was in Germany. Coming to different organizations related to seed and their establishment. AOSCA in 1919, CSTL during 1960, World Seed Year 1961, AICRP on maize 1957, NSC 1963, Seed Control Order 24th February 1983, ISST 1971, ISTA 1924, National Seed Project 1976. Basic seed cleaning is by air cleaner. Guard sample should be stored in seed testing lab for 1 year. Accelerated aging test is given by D. Lanche. Lowest seed multiplication rate is found in groundnut, that is 1:6 to 8. Now this has been revised. You just check it in the ICR website. Premature flowering in cole crops is by bolting. The different important varieties in different crops and their parentage. Sorghum CSH1, which is the first sorghum variety, parentage is CK60A into IS84. Sunflower BSH1 CMS 234A into RHA 274 is the parentage. In cotton, Varlakshmi parentage is Lakshmi into SB 289E, which is interspecific hybrid. H6 parentage is AK32 into DHY 286-1. OCH32, that is Jayalakshmi, parentage is DS28 into SB425. For tobacco, GTH1 is the popular hybrid. Castor GCH3. The parentage is TSP10R into J15 JS15. Then for Bajra HB1, parentage is TIFT23A into BIL3B. Coming to seed formulation and development. Archisporial cells are the special cells of nucellus which undergo cell division from megaspore mother cells. Here you just remember what are these cells. Okay, whenever some definition is given and the 4 options are given, you should be able to identify that. Archisporial cells means special cells of nucellus. Nucellus, as you know, it's a part of seed which undergoes cell division from megaspore mother cell. Okay, embryo sac where 3 antipodal cells having chalaza and 2 synergids and 1 egg which will be fused at micropylar, and 2 polar nuclei are at the central which will be having n plus n ploidy level. Microspore formation is with 2 mitotic division. Pollen will undergo mitosis, then 1 generative and 1 vegetative. Again mitosis, 2 generative will be formed. Megaspore formation or embryo sac formation, 3 mitosis takes place here. Megaspore mother cell formation after 3 mitotic, 8 cells and then 2 polar nuclear cells fuse to give 7 celled and 8 nuclei embryo sac. Coming to development of ovary or female part development. Developing ovule is attached to placenta by funicules. Nucellus, that is a nutritive tissue, supplies nutrient to embryo. In some nucellus, perisperm is found, example in cocoa, pepper and sugar beet. Outer seed coat outgrowth is caruncle, that is found in castor. Hard seed coat is found in Malvaceae, Leguminosae, Anacardiaceae. Aleurone layer is 3N, which is a storage hub or storage place for enzymes and proteins. Endosperm is absent in orchids. Embryo and aleurone layer is living. Endosperm is non-living. Endosperm and seed coat are non-living kind. Pollen enter through micropylar end is sporogamy or porogamy. From the polar end, if pollen enters, it is in case of chalazogamy found in casuarina. Pollen enter through funicle or integument means mesogamy. During seed development, endosperm develops first, then embryo develops. This is the function of endosperm is to store and supply for nutrients to the developing embryo. So first food storage will happen, then embryo development will take place. Pollen storage. Cold storage means -5 to 10 degrees Celsius at the rate of 50% humidity. That is cryopreservation, and other method is dry method also. Coming to the chemistry of seeds. Major form of carbohydrates in seed is hemicellulose and starch. Seed albumins are soluble in water. Example in case of pulses, it is leguminine. Leucocins in case of cereals. Arachine in case of groundnut. Globulin form is soluble in saline solution. For that example is groundnut. Then seed prolamines are soluble in ethyl alcohol. Example zein in maize and gliadin in wheat. Hypogeal germination where epicotyle elongates. Epigeal germination, hypocotyle elongation. Paddy can germinate even in the absence of oxygen. That means under anaerobic condition paddy grows. As you know, 5 centimeter water stagnation is always maintained in the paddy fields. So absence of O2 is also okay. Seed replacement ratio for self-pollinated crops, it is 33%, cross-pollinated 50% and hybrids 100%. In 1960, first maize hybrid released. 64, first sorghum and bajra hybrid released. In 1969, Tharai Development Corporation was established. This is all about part 3. We'll meet again in part